like antibodies against latent TB in bacterial system. I'm Yeon from Molecular Medicine, Mixed Mode Batch 7, and currently under supervision by Associate Professor Dr. Tai. Just a brief background of my study. I'm currently working on an infection state caused by Mycobacterium tuberculosis, known as latent TB infection. So by definition from WHO, latent TB infection is the persistent immune response towards MTB antigens without evidence of TB symptoms. So as definition it suggests, individuals with LTBI will appear normal and healthy in the social settings as they do not manifest symptoms of a TB. So why is it important to diagnose individuals with LTBI? And the answer lies in the passage that I found in WHO Consolidated Guidelines on TB Preventive Treatments, in which it stated 3 main points why is it important to diagnose individuals with LTBI. And one of it is being 1/4 of the world population are actually estimated to be infected with LTBI. And secondly, they have also stated that these individuals will have 5 to 10% lifetime risk of activating to active TB disease, stating that they are able to transmit this disease to another person and starting a new transmission chain. And last but not least, they have also highlighted the importance of screening for TB to rule out TB disease and also testing for TB infection, which is LTBI, in order to achieve the global targets of anti-TB strategy. So this state why diagnosing LTBI is important as to eliminate TB in the near future. Unfortunately, currently we do not have a diagnostic tool, gold standard diagnostic tool in LTBI, but we do have a widely used test in the clinical settings for LTBI, which includes tuberculin skin test, TST, and also interferon gamma release assays, IGRA. However, both of these tests comes with certain limitations as they do not provide a direct measurement of MTB antigens within the host and also do not differentiate between ATB and LTBI. This suggests a major limitation or a big gap to be filled in for the future improved version of the diagnostic tools in a LTBI diagnosis. One of the promising entity in LTBI diagnostic could be TCR-like antibodies. So from the literature review, it suggests TCR-like antibodies possess the ability from both cellular and humoral arms of our immune system, stating their ability to both recognize intracellular peptides that are presented on MHC molecules and also elicit the effector functions of an antibody. So from here, Mycobacterium tuberculosis is an intracellular antigen, pathogen, hence TCR-like antibodies is a suitable entity in diagnosing and treatment of LTBI. Previous study by my senior has already generated these TCR-like antibodies, namely HLA-A2, A11, and A24 through phage display technology. Apart from that, they have also get their functionality tested on the pMHC molecules, where the MHC molecules cover 70% of the Malaysian population. For the peptide molecules, it originates from a 16 kilodalton heat shock protein that was shown in literature review to be primarily produced only during MTB dormancy. This suggests that this peptide, by detecting this peptide, it has the potential to differentiate from LTBI from ATB, stating their diagnostic applicability. Hence, TCR-like single-domain antibodies in such case is a potential entity as it is able to mitigate the limitations of current diagnostic tools, which they now provide direct measurement of MTB antigens and also differentiate between ATB and LTBI. The focus of my study is to bridge the gap between antibodies generation and discovery part to the potential point of care diagnostic application in the biosensor development in the near future. So we are aiming to express these TCR-like antibodies in a fast and cheap way. Hence, this leads to the general objective of my study, which is to optimize the expression of A11 TCR-like single-domain antibodies in bacterial expression system, so just to prepare for a large-scale production in the near future. The specific objective of this study are as shown on these slides. So briefly, first objective, I have to do a molecular cloning, which is to clone the HLA-A11 TCR-like single-domain antibody genes in the suitable bacterial expression vector. Next, I have to optimize the bacterial expression and also protein purification conditions for this single-domain antibody expression. And last but not least, with this expressed protein, I have to confirm their functionality against their kinetic antigens with a dot plot. The overview of the methodologies are as shown on these slides. So briefly, for objective number 1 is just molecular cloning of this HLA-A11 TCR-like single-domain antibody gene into PET51B bacterial vector. For objective 2, I have to optimize the induction condition, which includes the temperature, IPTG concentration, incubation duration, and also to optimize the purification of this protein. And last but not least, I have to confirm the functionalities against their kinetic antigen with a dot plot. The progress and the results are as shown in the subsequent slides. Currently, I have already achieved my first objective, which is the molecular cloning part. For molecular cloning, I start off with a PCR reaction, which I first determine the annealing temperature of my primer, followed by a PCR reaction with a high fidelity polymerase Q5. With that PCR product, I perform a RE digestion with both NCO1 and NOD1 enzymes, and the target bands was separated using gel electrophoresis. And by then, the DNA bands were extracted through gel extraction, and the target DNA is ligated into a suitable plasmid, which is PET51B. Following that, this DNA ligation mixture was transformed, and subsequent day, the colony PCR was performed to preliminary confirm the success of a molecular cloning. And last but not least, we have sent the plasmid for sequencing and for a final confirmation. The gradient PCR results are as shown on these slides. So briefly, I have run a gradient PCR at a temperature ranging from 46.2 degrees Celsius to 58 degrees Celsius, which were loaded into well 1 to well 7 accordingly. The target gene that we are looking at is around 1298 base pairs, starting from the forward primer to the reverse primer, in which we can see a target gene band at around 1298 base pair in all wells from well 1 to well 7. The DNA band and size, DNA thickness and intensity do not differ much from each other. Hence, we are choosing a temperature of 50.2 degrees Celsius for the subsequent PCR reaction with high fidelity polymerase Q5. Upon performing the PCR reaction, we are also getting a band at 1298 base pairs. Hence, this PCR product was then proceed for a PCR cleanup, just to ensure that any buffers in PCR reaction would not interfere with the subsequent RE digestion. So upon PCR cleanup, the concentration measured is around 199.7 nanogram per microliter with a purity of 1.82. Following that, a double restriction enzyme digestion was performed with NOT1 and NCO1 enzymes. So briefly, what I did is I have loaded well 1 to well 4 with A11 single-domain antibody gene post-RE digestion as loaded into well 1 to well 4. For well 9 to well 12, I've loaded with PET51B vector, which was also double restricted enzyme digested. Here, the DNA bands that we are expecting, the size is around 389 base pairs for A11 single-domain antibody gene after restriction enzyme digestion, in which I have gotten this band at around 400 base pairs, as you can see on the gel upon gel electrophoresis. For PET51B, I have also gotten a band size at around 5131 base pairs, which is as expected. So both of these bands circled in red are cut out, and the DNAs are extracted from these bands, and the concentration was again measured, and the concentration of A11 single-domain antibody gene is at around 7.633 nanogram per microliter with a purity of 1.76, And the concentration of PET51B is at around 33.6 nanogram per microliter with a purity of 1.82. These DNAs are then ligated together at a vector to insert ratio of 1:1, 1:3, and 1:5. Following that, this ligation mixture was transformed into a bacterial cells. On the subsequent day, a colony PCR was performed just to preliminary confirm the cloning results. So what I did in the cloning is that I have inserted these 389 base pairs into a 5131 base pairs PET51B plasmid to get a total base size of 5520 base pairs. Now, the target band size that we are expecting to obtain in colony PCR is around 622 base pairs, starting from the T7 promoter forward primer to the T7 terminator reverse primer, containing the target genes. The results of agile electrophoresis for colony PCR are as shown on these slides. So briefly, what I did is that I've picked positive clones, 6 colonies from each vector to insert ratio culture plates, and then perform a colony PCR. So from well 2 to well 19 was loaded with different colonies from different vector to insert ratio. Here we can see that we are getting a band at expected 622 base pairs for some of these colonies. Those that are circled in red are the colonies that we have performed a plasmid extraction and the DNA was sent for sequencing. Upon DNA sequencing, a sequence alignment was performed, and we can see that the clone genes have 100% identity to the original sequence, as shown on the percent identity matrix, 100% for all of these genes. So then these clones are now proceed for protein expression optimization, which I'm working on currently. So for the Gantt chart, I'm expected to, I've started my research activity in 2024 October, and we are expecting to end at around September of 2025. Currently, I've achieved my first milestone, which is the first objective, cloning of target genes into a bacterial expression system. Currently, I'm working on the expression and purification of TCR-like antibodies in bacterial system, and I'm currently in line with the timeline that in the Gantt chart. So we are expecting to finish this project in the expected timeline. These are the references, and thank you very much.
Thank you for your nice presentation of explaining what you have done, Lian. Can you go to your slide 17? I just
17.
The point.
All right.
Okay, so like now you have said that you have got 389 base pairs, correct?
Yeah.
But at the same time, you have, I think, bands at 750 and 250. So what are that bands are?
Okay, so for this 750 base pair is this one, because the whole gene sequence that I've PCR out is this 1298 base pairs. So upon enzyme digestion with NCO1 and NOT1 enzymes, we are getting 3 bands actually is around 740 and 169 base pairs. So I think both these bands are from here.
Okay. So then how did you did your extraction? So what type of extractions that you do to get 389?
Sorry, come again, the question? To get what?
So how did you isolate your 389 base pair?
Oh, I did a gel extraction with a gel extraction kit. So briefly what I did was just, I cut out this band, and then later on I just use the kit to extract the DNA.
Okay. Can you go to the 6/8 something image, gel image?
this one, 622.
622.
Okay.
So the red color bar is the one that you selected. Am I
Yes,
correct?
correct.
Okay. Can I know that why you didn't select number 12?
Oh, number 12. Oh, initially, okay, to be honest, I want to select all those with the band, but DNA sequencing is expensive. So
Okay.
I just select those, I think, okay lah, got bands means can already.
Check out my
That is the
pocket. Check out my
reason.
pocket.
Because like if you ask me, you see number 8, number 8, number 5, number 12,
Yeah.
16, they have less primer dimers or non-specific binding down there. And then no, I'm just asking what is the reason that so you just randomly picked.
Yeah. To be honest, I really randomly pick because I feel like once there's a band there means it is we have already preliminary confirmed that this molecular cloning works. So yeah, I'm quite confident that it will turn out positive lah, the results for DNA sequencing.
Okay. So after your DNA sequencing, so what do you check for?
Also I sequence this region starting from the forward primer to the reverse terminator because this blue color region originates from the PET51B, including this blue color region will originate from this PET51B, and the orange color region will originate from the previous, the target gene that we are excising. So this confirms the DNA sequencing gene result.
So when sequencing, you check whether the sequence that is in place, correct?
Yes, correct.
Do you check for any mutations, anything, whether that is in frame or not?
Yes, it is in frame. And then I've also checked the mutations, and then because we have performed this we have performed this molecular cloning in a SNP gene, and then following that we have already after we have gotten these molecular cloning results from DNA sequencing, we compare it to the original sequencing upon with the SNP gene one, and then it is 100% identity. We are getting 100% identity for all the sequence.
I think you need to explain that what you have done. Because just showing the image of 100, 100, 100 is not.
Okay.
Tell me, as a person who did do the work, okay. So next time when you present, you must put out that what you look for.
Okay,
so
okay.
100% identity is what you're trying to mean, whether that is still in frame. 100% identity is the sequence, but when you put it in, that whether that it sits in frame or not. So you have checked all of that. Okay.
Okay.
You have done the work. It is just that how you present it. Because I didn't do the work, so I don't know. So you have to tell me what you've done.
Okay. I know that. Later on, I will present it not.
Yeah. Okay. Good luck. I think you have done quite a lot, and you have just a small bit more pieces to finish up, and your functionality test. Hope you will finish it before the time, and you have more time to write. Okay. Thank you, and good luck.
Thank you very much.
Dr. Liao, do you have questions?
Yeah, good morning, and thank you, Liam, for your great presentation, and have a good supervision from Dr. Tai on your project. I'm pretty sure that this project you're going to be success, but like what Dr. Venu mentioned, is you just need to present it in a better manner. Okay, I basically that I don't have much comment just on your introduction, and can you go back to your introduction, your title and your introduction?
Okay.
Okay, good. And
introduction. This one.
Yeah. All the introduction. I'm pretty sure that you have done a very good introduction, and this is you have provided.
Okay.
Yeah, I mean that all the introduction, because your title is on the expression of this TCR-like antibody against using the bacterial system, right?
Yeah.
And you have done pretty much on what's the function of TCR, everything, blah blah blah, and how good it is, and what. Okay, this is what I mentioned. So it's a very good, because you have a very good insight to the readers who are not in this field. But I find that in the introduction, you are not much touch on the expression system, like especially you are using the E. coli system. Okay. Yeah. Maybe in the next presentation, you can add one more part of why you want to use bacterial system and what is the benefit, why you want to choose this system to express. This is your
Okay.
core work, right? This is your core.
Yeah.
So for those like TCR, everything, all those kind of thing, and you still can touch it, but can minimize it and focus on your expression system. When you can compare it compared to other expression system, why is the benefit of using the bacterial system? Because this is your work.
Okay.
All right. So this is my suggestion,
Yeah.
Ian. Basically,
Yeah.
it's done really well. Okay, very nice diagram, everything. All right. Can you go following like what Dr. Wano mentioned about the slide in 20? So I saw 20 just now. You say because your supervisor you want to jaga his pocket, and so we can understand that this is all the supervisor facing right now. And so if I were you, I think that I will choose the clone, like for example, clone
Number 12.
number 12. Yeah,
Yeah.
for this. So I wouldn't say I don't go for the clone that you selected, just because sometimes you can see those kind of diamond happen, and we try to skip it. So so this, I think you have answered it just now, and maybe this one you also next time you have to bear in mind, like when you want to choose the clone, and of course that the diamond it wouldn't show that it can be any happen, but we try to minimize the possibility of any sequencing error. Okay, so we'll go for the specific clone.
Okay.
All right, then go to slide 21, please.
21.
So I think that just now, because there are so many hundred here, like the original, I'm pretty sure that this is your positive control clone, right?
Yeah.
Yeah, positive control, and yeah, perhaps that you can label it up there saying that this is your post control. Then you just want to compare your clones 2 to 7 to your post control, and so what is the 100 in the lane 1, lane 2, lane 3, lane 2 until the lane 6? So you need to label it. Okay.
Okay.
In conventional way, how did we present the cluster W is you need to put a sequence, but if some of the sequence maybe is not not to be ready to disclose, and then you can use the star those kind of coding and then to cover a little bit. It's fine, but at least you need to show that. Okay, you can change that. Yeah.
Okay.
There's a tools. I don't know what's the name already. Not biorender. Cluster W1, and you can check with your supervisor.
Okay.
It's a very common. That tool is very common. You can change it. Right.
Okay.
Can you go to the next slide, please?
Next slide. Gantt chart.
No. Previous. Sorry. Previous.
Previous slide. Okay. This one.
Previous again. Okay. Now I like to learn from you lah. Okay. Why you want to choose this PET 51B? There are quite a lot of the vector, and why do you want to choose this PET, this expressions vector?
Because first of all, this is the currently available vector in the lab. Secondly, we are trying to this PET51B vector carries this his tag. And we are trying to purify this protein through his tag, and this his tag might be useful for the downstream biosensor development as well. So now we choose PET51B.
Okay, so in which E. coli do you plan to transform it into?
Currently, I have already transformed into a T7 shuffle cells, bacterial cell.
Why do you choose T7 shuffle cells?
Because T7, according to literature review, T7 shuffle cells has the ability to form a good disulfide bonds.
Okay, and I think there are also a few of the E. coli, right? For example, Rosetta gummy, this kind of E. coli. Have you studied it? Have you of course, you don't necessarily to try it in practical way, but have you studied, do any comparison? What is the benefit of this T7 compared to Rosetta gummy?
Okay, I do not know about Rosetta gummy. I've read other bacteria also, like BL21 and others. I mean, not Rosetta gummy.
Yeah. But it's okay. Sorry. But this one maybe you need to, because your work is more focused on the expression, and this is a lead review, and like just now I mentioned to you, you need to do some comparison because you choose the PET 51B and T7 as your target of the clone and expression vehicle. I mean the T7 as well. So other kind of the vehicle, even you are not doing on that, perhaps you also need to understand what is the function of each of them in your disease maybe.
Okay.
This is my little suggestion only. Because it's very well, of course, I can say that your presentations, you did very well, prepare all the slides very clear, but only like you need to put more focus on your objective, like for because you're working on the expression.
Okay.
All right. Thank you so much, Ian, and good luck to you. Thank you so much, Bung Lucy. I'm done. Thank you.
Thank you, Dr. Liao and Dr. Venu. Any other questions from the floor? All right. So thank you, Ian.
Okay, thank you very much,
Thank
everyone.
you. Thank you.
Thank you, everybody. Thank you.
Dr. Tai. Dr. Tai, you are going to chair the session. So I pass it to you.
No, I have a meeting now. So Dr. Lin has kindly agreed to chair the next meeting. Thank you.
No problem. So maybe you can upload the slides. Can you upload your slides now?
Yeah.
Forgot to speak slowly. Can you upload your slides now?